what do we learn in this chapter specifically? What is this chapter about? How do you think living organisms control all the functions? You know that one is called as the nervous system, second one is the endocrine system. That is in the higher organisms. So what is the role of DNA then? What is the role of nucleic acids? There are four types of biomolecules in the living system, right? What are those? Carbohydrates, proteins, lipids, and nucleic acids. So carbohydrates, lipids, proteins, we know their function, at least some. Like proteins function as enzymes, lipids make a plasma membrane, and they also form the storage materials, correct? What is the function of nucleic acids? So in the textbook, there is something mentioned like this. Central dogma of molecular biology proposed by Francis Crick, one of the scientists who discovered the structure of the DNA. What does it say here, guys? Something showing as DNA to mRNA, mRNA to protein. What is that? What exactly the DNA is made up of? Okay, there are two types of nucleic acids. We'll come back to this later. I think you have no idea about this one. Now, fine, you see, guys. There are two types of nucleic acids. One is a DNA, other one is a RNA. So, you see here. These are the two types of nucleic acids found in the living systems. Two types of nucleic acids. DNA acts as genetic material in most of the organisms because some may have the RNA as genetic material. RNA, though, it acts as genetic material in some viruses like TMV, QB bacteriophage, but mostly functions as a messenger, adapter, structural, and catalytic molecule. So we can say, we will say that DNA is a major genetic material in the most of the organisms. What is meant by the word genetic material? It carries the genes. What are the genes? What are genes? Genes are the bits of DNA which carry the information for the synthesis of the proteins. Okay, you see here. See, different organisms have different lengths of the DNA. You see here. DNA is a long polymer of small units called the deoxyribonucleotides. The length of DNA usually defined as the number of nucleotides is also characteristic of an organism. So, one organism may have 1 million nucleotides, some other organism may have a billion nucleotides. Now, it differs from organism to organism. For example, bacteriophage Cyclos 174, what is the number of nucleotides here? 5386. Bacteriophage lambda, what is meant by bacteriophage? What is a bacteriophage? A virus that infects bacteria, correct? It has 48,502 base pairs. E. coli has 4.6 into the power of 6 base pairs, and human haploid, no, not diploid, haploid is 3.3 into 10 to the power of 9 nucleotides. So all these nucleotides, they join together, correct, through with some chemical bonds, and they form the genes. You see here, if you take a structure of a polynucleotide chain, where there is a DNA or RNA, they are the polymer of nucleotides. Each nucleotide has how many components, guys? 3 components. What are those? A nitrogenous base, a pentose sugar, and a phosphate group. The nitrogenous group is of two types, purines and pyrimidines. What are the examples of purines? Adenine and guanine. Pyrimidines? Thymine, uracil, and cytosine. And the pentose sugar, what is meant by the word pentose sugar? It has 5 carbons. Yes, 5 carbons. It is a monosaccharide. And there are two types of sugars found in the nucleic acids. One is called as the ribose, that's where the name ribonucleic acid. Deoxyribose, that is especially the second group, second carbon of the second carbon atom of the pentose sugar in the DNA as a only hydrogen. That means it is called as the deoxyribose. You see, purines are same in RNA and DNA, which is cytosine is a common to both DNA and RNA. What is the difference in the nitrogenous bases between RNA and DNA? DNA contains adenine, thymine, guanine and cytosine, whereas the RNA contains adenine, guanine, cytosine and uracil. That's the difference. What's the other difference you can say between the DNA and RNA? DNA is mostly double stranded, RNA is single stranded, correct? Now, let's see. What are the links? What are the chemical bonds found in the DNA? How does What is the chemical bond between a nitrogenous base and a pentose sugar? No, it's called N-glycosidic bond. What is the chemical bond between the pentose and the phosphate group? Ester bond, correct? So they are linked together. And what is the bond that connects the two nucleotides? What is the bond? It's called phosphodiester bond. Now, then let's say one polynucleotide chain is made up of nucleotides linked together the phosphodiester bonds. Now, in DNA there are two chains, correct? What is the chemical bond that joins between these two chains? They call as the hydrogen bonds. They call as the hydrogen bonds. So, adenine base pairs with thymine with two hydrogen bonds. Guanine base pairs with cytosine with three hydrogen bonds. Understood, right? That's the gist of this one. Now, see here. So, DNA, we call it as a deoxyribonucleic acid, which means it's an acidic substance, correct? Now, the first person to identify the nucleic DNA as what to say DNA as a substance is a Frederick Michel. In which year, guys? 1969, and he called it as a nuclein. Now, two more scientists, Maurice Wilkins and Rosalind Franklin, together they used a study called as the X-ray diffraction to give an idea that DNA is a double helix, which was completely elucidated by two more scientists. One is called as James Watson, other one is a Francis Crick. In which year? 1953. So what are the features of this one? You see, guys, simple. There's one more scientist called as Erwin Chergaff. He gave the rules of base pairing. He said that adenine base pairs with thymine, guanine base pairs with cytosine. And he also said that number of purines in a DNA molecule equals to number of pyrimidines. If adenine is there, there will always be thymine. Adenine is, guanine is there, then there will be cytosine on the other strand, correct? You see. And this base pairing is always complementary, a unique property to the polynucleotide chain. What is meant by complementarity here? What is complementarity? Are both the chains same or different? What is it, guys? Complementarity means, like I said, adenine base pairs with thymine, guanine base pairs with cytosine. If one chain has purine, the other chain will have a pyrimidine. This is called as the complementarity, right? Now, anyway, now the sentence mentioned here. If each strand of parental DNA acts as a template for synthesis of a new strand, the two double-stranded daughter DNA molecules produced would be identical to the parental DNA molecule. Now look at this. This diagram is not mentioned here, you see. Let's say this is a DNA molecule. There's a parental DNA. There's a parental DNA. Now it is replicating. So this is one strand. Let's call it as a template 1. There is also another strand which is a template 2. Now every time when a cell divides, its DNA also doubles, right? So each strand functions like a template. Now there is a one parental strand. This is one more parental strand. Now, when the new DNA molecule is synthesized, a new strand is added to this one. A new strand is added to this one. You see guys, this is a template one, right? The new strand which is synthesized is complementary to the template, which means these two strands are same as a parental DNA. Are you following my point? For example, if this strand has adenine on this side, the other strand will have, what is it have guys? Thymine. If this strand has guanine, the other strand will have the cytosine. The same thing can be seen in the parental DNA. So this process of synthesizing a new DNA molecule is called as a, what is called? Replication. And this replication, which is proposed by Watson and Crick, it is known as the semi-conservative mechanism. It's called as a semi-conservative mechanism. Just give me a moment. So, what about the RNA? RNA has the same rules or different rules about the complementarity. Is there a complementarity in the RNA molecule? No. Why not? Because it is a single stranded molecule. Understood? Right? Let's see. Just give me a moment. Okay. See here. So, what are the salient features of a DNA molecule? It's made up of two polynucleotide chains where the backbone is constituted by sugar, phosphate, and the bases project inside. What is meant by this sentence? Anybody? Anybody about this sentence? Let's see, guys. If we draw the DNA like a ladder, So this is one strand, and this is one more strand. So this strand is called as a backbone. So this backbone has a sugar and phosphate, sugar, phosphate. This one also has sugar, phosphate, sugar and phosphate, which forms the, let's guess here, the backbone of the DNA. Whereas the nucleotide bases, they project inside. Let's say there is a nucleotide base, sorry. It's a nucleotide base. That means the core of the DNA is made up of, what's guys here? Nitrogenous bases. Whereas a backbone is formed by the sugar and phosphates. That is the meaning of the sentence. Next you see. What's wrong with this one? Okay, right. 95. Okay. The two chains of the DNA are anti-parallel to each other, which means if one chain runs in 5' to 3' direction, the other chain runs in 3' to 5' direction. So what do we have on the third carbon atom of the sugar? A hydroxyl group. And the 5' carbon out of the sugar is, what does it contain? Phosphate. So one side is hydroxyl group, other side is the phosphate group. Another chain, it is opposite. This one will have hydroxyl group, the other one will have the phosphate groups. So two chains are anti-parallel to each other. 3' to 5', 5' to 3', right? Next. Okay, we said this already, the hydrogen bonds, and the two chains are coiled each other in a right-handed fashion. Then, this is very important. The pitch of the helix is 3.4 nanometers. What does it mean by each one turn? You can see, guys. So this is one complete turn from here to here. One complete turn has how many base pairs? 10 base pairs. And the distance between the two base pairs is approximately 0.34 nanometers. One length of one complete helix is called as how much is it? 3.4. And how many base pairs accommodated in one turn? 10 base pairs. That means the distance between the two base pairs is 0.34 nanometers. Right? Then, okay, let's see here. Coming back to this part here. So when we say the DNA is a genetic material, it contains the nucleotides in the form of the genes. And these genes are transcribed into different types of RNAs. There is mRNA, there is tRNA, and then there is rRNA. So earlier we mentioned one point here. RNAs can function like, what's the guess here? In some, they act as genetic material. In some organisms, they function as messengers. What's the meaning of the word messenger? They transmit the information from the DNA, and this information is used to synthesize the proteins. Second one is adapter. What is an adapter here? The tRNA molecule. What is the function of tRNA molecule? What's the function? You learned this chapter very recently, right? Even you wrote the exams, not even a single word coming out of your mouths. It transfers amino acids for protein synthesis in the ribosomes. What is this one here? Structural. Ribosomes. What are ribosomes made of? What ribosomes are made up of? Next. Did you read the chapter before coming here? Guys, I'm asking you only. Why not? Miss, what about you? What ribosome is made up of? Ribosome is made up of two components, rRNAs and proteins. That's why they are also called as the ribonucleoproteins. Means RNA can also form the structural components. And third, last one is catalytic. RNA molecules can function like enzymes, which are called as the ribozymes. Correct? So RNAs have 5 different functions. They can act as genetic material, they can act as messengers, they can act as adapters, structural components, and also catalytic molecules. So because of this reason, the DNA dependent on the RNA molecules to transfer the information to produce the proteins. So the information in the DNA is present in the form of the nucleotides. What are those nucleotides? Deoxyribonucleotides. What are the nucleotides found in the RNA? Come on, tell me, what is that? Ribonucleotides. So there is a transfer of script from deoxyribonucleotides into ribonucleotides. So that process is called as the transcription. And the RNA, let's say the mRNA, once it's synthesized in the nucleus of eukaryotic organisms, it will come out and bind with the ribosomes, and this information present on the RNAs is used to synthesize the proteins. The information present on the mRNA is in the form of the ribonucleotides, whereas the information present in the protein, not information, the component of the protein present is in the form of the amino acids. Means the language has been translated from nucleotides into amino acids. So this process is called as a translation. And then it's mentioned in some viruses, the flow of information is in opposite direction, that is from RNA to DNA. In majority of organisms, the information is transmitted from DNA to RNA to RNA to proteins. If this goes in opposite direction, it is called as a reverse transcription, or reverse of central dogma. Are we clear there? Right. Now you see. So this is how the DNA is packed in the organisms. See, for example, in a typical mammalian cell, let's say in humans, the DNA double helix has a length of 6.6 into 10 to the power of 9 base pairs. This is a long molecule actually, and the length is approximately 2.2 meters. This is how you can calculate, which is far greater than the dimension of a typical nucleus. The nucleus has a diameter of how much, guys? 10 to the power of minus 6 meters. But the length of DNA is about 2.2 meters. Now, this DNA is packed in the form of the chromosomes, and those chromosomes are are made up of small units known as the nucleosomes. And this packaging is helped by a special type of positively charged basic proteins, which are called as the histones. So histones are like 8 histones together, they join together, and the DNA wraps around this histone octamer, which is called as the nucleosome. And each nucleosome has a length of approximately 200 base pairs. How much is it, guys? 200 base pairs. And these nucleosomes, it's like they wrap around each other, forming more compact structures, which are called as the chromosomes. Or you can say chromosomes can be visible only during the cell division, but during the rest of the time, the DNA is present in the form of the, what is called, guys? Chromatin. And there are two types of chromatins. One is a euchromatin, other one is a heterochromatin. What is euchromatin here? It's loosely packed, and when you stain with a specific dyes, it appears light and which is transcriptionally active, which means the DNA which is used to synthesize the proteins is always in the form of the euchromatin. The DNA which is not at the time useful, it is compactly packed, which is called as the, which is densely packed, stains dark, and transcriptionally inactive. Am I clear about this? Right? And then there's a point mentioned here, you see, guys. RNA world. RNA was the first genetic material. When living systems were evolving, the first unstable genetic material to form was RNA. Later on, a more stable form has evolved, which is called as the DNA. You see here, the essential life processes like metabolism, translation, splicing evolved around the RNA. Because you see, living systems still use the RNA to transfer the information from the DNA to produce the proteins. They have to go through a process called as the splicing, capping, splicing everything, and also the translation and the metabolism. RNA used to act as genetic material as well as a catalyst. Still, in the ribosomes, RNAs are there which can function like the enzymes called as the ribozymes, which can form the peptide bonds. So, it was reactive and is highly, and the problem with RNA is it was highly reactive, right? And it is unstable because of the hydroxyl group on its second carbon atom on the pentose sugar. Therefore, DNA has evolved from the RNA with chemical modifications that make it more stable. DNA can survive for generations together, correct? But RNAs, they can, once the function is done, they will be, let's say, what they can call them as a recycled, correct? Now, so in this part, we know that this is how the information is passed on from DNA to RNA to the proteins. Now, scientists were skeptical what is the actual genetic material. They knew that there are 4 biomolecules. Carbohydrates are there, proteins are there, lipids are there, and nucleic acids. To confirm that DNA is genetic material, several scientists perform many experiments. The most common experiment, one is the Griffith's experiment. Second one is Avery, McLeod, and McCarthy's experiment. And third one is Hershey and Chase experiment. Okay, what Griffith did actually, tell me guys, what was Griffith's contribution in finding out the DNA genetic material. You have to remember here, he used a bacterial strain called as the Streptococcus pneumoniae, which causes, what does it cause, guys? A disease, right? What disease is it? Pneumonia, right? So he used two types of strains. One is called as the rough strain, other one is called as the smooth strain. Rough strain causes pneumonia or not? You sure? I say here, some produce the shiny colonies while others produce the rough colonies. Their strain kills mice while our strain do not develop. Okay, so the smooth forming colonies, bacteria can cause infection. As a rough strain do not cause infection, right? You see here. What he did was a very simple experiment, guys. He injected the R strain into the mice, and another group of mice injected with the S strain. Which one was killed here? The one that was injected with the S strain, right? That is done. In the second experiment, he killed the rough strain and injected into the mice. What happened? Was it a disease? No. So what he did next? He mixed the heat-killed smooth strain and the live R strain together. So when injected into the mice has been killed. So what does it suggest here? Something happened here. Something has been transformed or transferred from the rough strain, so from the smooth strain to the S strain. So we had no idea what it was actually. So came one more experiment which is done by the Avery, McLeod, who did several experiments and found that it was the DNA which has been transferred from one strain to the other strain. But it wasn't enough. We need a concrete proof that it was the DNA which is the genetic material. Comes the next experiment performed by Hershey and Chase. So they used bacteriophage and E. coli bacterial strains. So finally, after this experiment, it was conclusively proven that it is the DNA that is genetic material, not the RNA, not the carbohydrates, not the proteins, not the lipids. Okay, you have to go through this clearly later. Now, is it clear, everybody? Okay, we discussed these points already. Now, so this is experimental proof of the DNA replication. So once the DNA structure has been elucidated by Watson and Crick, they also proposed how the DNA doubles itself, which is called as the replication. And this, what did they mention? Watson and Crick has immediately proposed a scheme for DNA replication while proposing the double helix structure of the DNA, where in the replication, the two strands of the DNA get separate and act as a template for the synthesis of new complementary strands, like I mentioned in the diagram before. Now, after completion of replication, the DNA molecule would have one parental strand and one newly synthesized strand. So, which suggests that the newly formed DNA molecule will have one old strand and one new strand, which is called as the semi-conservative mechanism of replication. And this was experimentally proven by two scientists who is called as Messerson and Stahl. So they used different types of, the E. coli bacterial strain, and they used radioisotopic molecules to find this experiment. Okay, if you have a doubt, you can ask me, or we can continue the next one. Is it fine? Shall I explain this one? Yes, explain. Okay, you see, guys, simple. Okay, we'll draw from here only. Okay, you see, guys. So the experimental organism here is a E. coli. What they did, they have grown E. coli in a medium containing the heavy nitrogen isotope known as the N15 for several generations. So after many generations, every nitrogen atom in the E. coli contains N15, including the DNA molecule. So when they performed, what do you say, the centrifugation and isolated the DNA, they found that the DNA of this bacteria contains N15. N15. So all the nitrogen molecules of the DNA contain N15. So next what they did, after a few generations, they have grown the bacteria in a medium containing N14. So what happens when the bacteria is dividing, it requires a new nitrogen source. The new nitrogen source is nothing but N14. So this has been continued for one generation. What happened then? The new DNA molecule, which has been isolated from the second generation, contains a hybrid of N15 as well as the N14. The parental strand is N15. The daughter strand is N14, which suggests that the old or the parental strand containing N15, the daughter strand contains the N14, which means the new DNA molecule formed contains one parental strand and one daughter strand containing two different nitrogen sources. And this has been continued for the next generation. 40 minutes means 2 generations, because E. coli duplicates for every 20 minutes. In the next, after 2 generations, what happened, you see guys? What do you notice here? The two molecules that are formed after 2 generations contain completely N14, and the parental strand is still continued to contain one N15 and one N14. So every time this experiment was conducted, they had the same results. One strand containing N15, other strand containing N14, which means the newly synthesized strand always contains one parental and one new strand, which is called as the semi-conservative mechanism. Understood? Right? Okay. Now, then, so what is the mechanism of DNA replication, guys? You see here. The machinery and the enzymes. E. coli completes replication in 18 minutes. The average rate of polymerization is about 2000 base pairs per second. Means every second 2000 base pairs will be added by the enzyme DNA polymerase. Energetically, replication is a very expensive process because it requires lots and lots of triphosphate molecules. Then there are 2 strands here, correct? DNA is always made up of 2 strands. One goes in 5' to 3' direction, other one 3' to 5'. And remember here one point very important. The synthesis of a nucleotide, so synthesis of a nucleic acid always is in 5' to 3' direction, because the DNA polymerase can add new phosphodiester bonds only in 5' to 3' direction. So because of this small glitch, the replication always occurs in a continuous process on one strand and discontinuous on the other strand. So here you see the enzyme which is required to perform the replication is called as the DNA dependent DNA polymerase. It uses the DNA as a template and it polymerizes the nucleotides. That's why the name DNA dependent DNA polymerase, which catalyze the polymerization process in 5' to 3' direction. So one strand with the template polarity of 3' to 5', replication is continuous, while on the other with the template polarity of 5' to 3' will be discontinuous. And the discontinuously synthesized fragments, which are called as the Okazaki fragments, they are joined together by an enzyme called as the DNA ligase. What is the function of polymerase? To synthesize a new strand. What is the function of DNA ligase? To join the fragments formed on the discontinuous strand. Now every replication starts from a point whether it is a bacterial cell or it is a eukaryotic cell. The point where the replication starts is called as the origin of replication. In prokaryotes, is usually one origin of replication. In eukaryotes, where the length of DNA is very long, they have multiple origins of replication. And during, okay, this is fine. This point. In eukaryotes, replication of DNA takes place in the S phase. I think you know cell cycle cell division, right? How many phases are there in this one, guys? How many phases are there in cell cycle cell division? S phase, M phase, and G. Right? S phase means the synthetic phase, where the required procedures are, I would say, the required things are made during the S phase. Okay, fine. Now, that is the first step in the central dogma. What is the second step, guys? It's a transcription. So, what is meant by transcription? The transfer of information from the DNA into the RNA. It is the process of copying genetic information from one strand of DNA into RNA, correct? You see here. So, this is the simple transcription unit of a DNA on the DNA. It has two strands, 3' to 5' to 3' and 3' to 5'. So, do you think both the strands are used as a templates here? No, only one of them. The one which runs here, 5' to 3' is a template strand. And since this occurs from the promoter. So, each transcription unit has three components. One is called as the promoter, where the transcription starts, and the last one is called as the terminator, where the transcription ends. So, the sequence between the promoter and the terminator is called as the structural gene. So, this is the sequence which is used to synthesize the RNA, right? You see here. So, during transcription, even the principles of complementarity governs or operates the transcription, except adenosine forms pair with the uracil instead of the thymine. You know that DNA contains adenine, thymine, guanine and cytosine, whereas RNA contains adenine, uracil, guanine and cytosine. So, instead of thymine, the RNA will have the uracil, right? So, each, you see guys, each unit has three components, a promoter, a structural gene and a terminator. And the enzyme that is used for transcription is called as the DNA dependent RNA polymerase. Enzyme for replication, DNA dependent DNA polymerase, which catalyze the polymerization in only one direction. What is the direction here? 5' to 3', which means which strand is used? Strand that is running from 3' to 5'. That is called as the template strand. The opposite strand is called as the coding strand. The strand that has polarity 3' to 5' act as a template, whereas other strand with polarity 5' to 3' direction and the sequence same as the RNA. Why it is same as the RNA? Because the strand which is used as a template is complementary to the coding strand, right? Now, so what is the location of promoter? It is always on the, where is guys? 5' end, that is the upstream of the structural gene, which we are referring to the coding strand. This is very important. These points are very important, guys. Next, it provides binding site for the RNA polymerase. So, the polymerase has to bind to a particular point, a particular sequence in the DNA, which is called as a promoter. And the terminator located towards the 3' end downstream of the coding strand, which defines the end of transcription. So, when the polymerase comes to certain codons, which are called stop codons, as they do not, represent any amino acid, that particular point is called as the end of transcription, or that sequence is called as the terminator. Are we clear there? Right? Now, To understand this one, we need to know something here. Okay, this is fine. Okay, this one. So genes are located on the DNA, which is functional unit of inheritance, which means it carries the genes which are transferred from one generation to the next generation. DNA sequence coding for tRNA or RNA are also define a gene. So generally we think that a gene is always the sequence of DNA which is used to synthesize the protein. It is not always the same, because a gene which codes for tRNA or RNA is also called as a gene. Gene doesn't have to necessarily code for a protein. It could code for rRNA, it could code for tRNA as well. Now, cistron is a segment on the DNA that is coding for a polypeptide. So, cistron is a word used for the gene which has a sequence to synthesize a protein. And there are two types of RNA molecules, mRNA molecules. Some of them are called as the monocistronic, some of them are called as the polycistronic. So, monocistronic genes are found in eukaryotes, polycistronic are found in the bacteria or other prokaryotes. Now, the eukaryotic genes have two types of sequences. Each gene has two sequences. Some of them are called as the exons, the other one called as the introns. So, which ones are actually used inside the protein? The ones which are called as the exons. Introns are being removed in a process called as the splicing, correct? Now, which is called as the post-transcriptional modifications, that will come back that. Now, where is this one? Okay, you see here. Only one type of enzyme called as a DNA dependent RNA polymerase catalyzes the transcription of all the three types of RNA molecules, mRNA, tRNA and RNA in case of bacteria. In eukaryotes, different types of polymerase are there. RNA polymerase binds to promoter and initiates transcription. As soon as the enzyme binds to the promoter, the transcription will start. It uses the nucleoside triphosphates. What are the substrates for the DNA-dependent DNA polymerase? The substrates for this enzyme is nucleoside triphosphates. What are the substrates for the DNA polymerase? Deoxyribonucleoside triphosphates. These are ribonucleoside triphosphates. Okay, now, so these substrates are polymerized by the enzyme in a template dependent fashion following the rules of complement. So they use a template. According to the sequence in the template, they will add the nucleotides to the growing polypeptide chain in the form of the RNA by using the nucleoside triphosphates. Now, only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA on the RNA polymerase falls off when they reach the part called as the terminator. After that, the RNA goes through, what is that? 3 steps. One is called as the capping, second one splicing, and third one is the polyadenylation, means addition of adenine molecules at the 3' end of the RNA. So once the introns are removed, all the exons are bound together to form a functional RNA called as the messenger RNA. Right? Now, next one here. Okay, it's done. What is this one, guys, here? What does genetic code means? So we know genetic information present on the DNA. What is genetic code? How does the information present on the DNA and the RNA is used to synthesize the protein? How it is done? How it is done? how does the enzymes, the machinery read the information on the DNA? Okay, we have alphabets, right? So we have the nucleotides, adenine, thymine, guanine, cytosine. How does these four nucleotides can code for 20 amino acids, 20 standard amino acids found in all the proteins? So when we arrange the alphabets in a particular manner, we form the words, right? Similarly, when these nucleotides are arranged in a particular fashion or the manner, they form the codes. And these codes are simply known as the genetic code. And genetic code, you see, which was, several scientists were involved here. One is Hargobind Khurana, second one is Marshall Nirenberg, and third one is Shihiro Okawa, right? Now you see, guys, these are the salient features of genetic code. The first one, the code is always triplet. For example, AUG is the first codon on the every mRNA molecule, which codes for amino acid called as the methionine. 61 codons for amino acids and 3 codons are stop codons. So there's something mentioned in the textbook. If, what they thought first, the codon may be single, codon may be doublet, or codon may be triplet. The triplet actually satisfies all the amino acid sequences. So when we multiply the codons, we totally get how many codons, guys? 64 codons. In that 61 code for amino acids and 3 codes for stop codons. What are the stop codons? Check the textbook. What are the stop codons? Right, UAA, UGA and UAH. These are the stop codons. The code is degenerate, means some amino acids are coded by more than one codon. So we have 61 codons, but how many amino acids are there? 20. Means some amino acids are coded by more than one triplet. And that particular feature is called as the degenerate, or the degeneracy of the genetic code. Next one. The codons are read on mRNA in a continuous fashion. Means there are no punctuations. When we write the genetic code, we do not use any kind of punctuations. It's a continuous sequence. The code is nearly universal. Means the for example, UUU codes for phenylalanine amino acid from bacteria to humans. That means every living system uses same type of genetic code. That's what it is called as universal. Of course, there are some exceptions. AUG has a dual function. It codes for amino acid. Methionine also acts as the starting codon or the initiator codon. UAG, UAA and UGA are the stop codons, are the terminator codons. Now the next part here is the translation. Okay, mutations is there, fine. What is a mutation? Simple, in 1 or 2 words, guys. Can someone tell me what is a mutation? A change in the genetic code, correct? Insertion or deletion of 1 or 2 base changes the reading frame from the point of insertion or deletion are called as the frame shift mutations. For example, you see, guys, okay. Okay, so these are the 5 codons here. How many codons? 5 codons. So this is the first codon, code for amino acid, methionine. Second codon, methionine. Third codon, methionine. Fourth one also methionine. Fifth one also methionine. Now you insert something here. Let's say instead of G, you have added U. What happens there, guys? It doesn't code for methionine, right? It codes for something else. Or you remove the complete sequence here. You remove the G. the first codon will become, what is it? AUA. Removing a nucleotide or adding a new nucleotide will change the complete sequence from the point of its insertion or the deletion. Those kinds of mutations called as what are called as frame shift mutations. And insertion or deletion of 3 or its multiple base, insert or delete in one of multiple codon, hence one or multiple amino acids will change. And reading frame remains unaltered from the point onwards. For example, let's say here. So these are all the sequence AUG, AUG, let's say. Now we have changed here. The frame before the insertion or deletion will be same, but the frame afterwards will change. Is it right? Why are you sleeping? You're sleeping. You understand the point here? Everybody? There's a sentence in the textbook mentioned. Open that part here. This section. Everybody? Open this one. Can you see there? There's a sentence called Ram has red cap. There was later added B. What happened? The first two words are same. The second two words are changed. In the next sentence, something has been added. Ram has, let's say, big red cap. So the insertion, the part before the insertion remains same, but the part after insertion will change. These are called as the different types of mutations, the frame shift mutations, insertions and deletions. Now, we'll come back to this. Right. The third step in the central dogma of molecular biology, the translation, which refers to the polymerization of amino acids to form a polypeptide. So, information present on the DNA has been transcribed into mRNA. The information present on the mRNA is used by the ribosomes and the other enzymes to produce a polypeptide chain or a protein. Now, the order and sequence of amino acids are defined by the sequence of bases in the mRNA. So, whatever the sequence present on the mRNA will be used and a new amino acid is added according to that particular codon. In the first phase, amino acids are activated in the presence of ATP, and that process called as the amino acylation of the tRNA. So, what exactly a tRNA, guys? Tell me. tRNA is also called as the transfer RNA. I told you earlier, RNAs can be messengers, they can be adapters, they can be catalytic, and they can be structural components. The tRNA is an adapter molecule. tRNA brings the amino acids in the ribosomes where they are added to the newly growing polypeptide chain. Now, this tRNA exists in a shape which is called, what is the shape called, guys? It looks like a inverted L in the two-dimensional. In the three-dimensional, it looks like a clover leaf. Looks like a clover leaf, right? Now, I'm not going to discuss these details here. So, let's say the first step is the binding of amino acid to the tRNA molecule. That is done. The ribosomes consist of structural RNAs and about 80 different proteins. So, one more RNA which is functioning like a structural component, that is a rRNA molecule. That's why it's called ribosomal RNA. It has two subunits, a larger subunit and a smaller subunit. The small subunit encounters mRNA, the process of translation begins. So, what happens here is the mRNA that has been synthesized in the nucleus, it comes out and binds with the smaller subunit of the mRNA, the ribosome, and immediately the larger subunit binds to it. Now, the ribosome slowly slides along the mRNA and the tRNA molecules reads the information on the RNA and adds a new amino acid according to it. And the first codon in the mRNA molecule will be usually the AUG. Is a AUG. You see here. See guys, this is a smaller subunit, it's a larger subunit. So mRNA is here, right? So this is the mRNA, and here the first tRNA molecule binds here. This tRNA molecule is bound to an amino acid. So this is a starting codon, let's say AUG. This is a stop codon, UAG. So first tRNA comes here, according to the codon, that is AUG. Now this is bringing an amino acid called as the methionine. Let's say the second codon, can you give me another codon something? An amino acid coded by lysine, what is it called? Lysine. Which amino acid codes the lysine? The genetic code. You have the table, right? Anything. Serine is UCU, right? The next one is UCU. Tell me one more codon. Huh? What is that? Lysine. Okay, that's the 3 amino acids. So this is the first tRNA molecule. Then comes the second tRNA molecule carrying one more amino acid. Now, a peptide bond is formed between these two amino acids. Now, after that, the ribosome moves one codon forward. It moves on one codon forward. Now, this first tRNA will be eliminated. Now, let's say this is gone. This consider the second one. This is a second one here. Bond with the 2 amino acids. One more tRNA comes here, bringing one more amino acid. One more peptide bond is formed. The ribosome moves forward. Now it is exposing the 4th codon, and this tRNA will be removed. So this same process continues until the ribosome encounters the codon AUG. By the time the ribosome comes here, the tRNA is carrying the polypeptide chain containing the number of amino acids similar to the number of codons. Once the process completed, the stop codon has encountered. What's wrong with this one, guys? See. So this is a diagram we have in the textbook. You can see clearly here, this is a growing polypeptide chain present or connected to one tRNA. So one more tRNA comes in here, forming a new peptide bond. So once this ribosome moves towards the last codon, that is the stop codon, the transition will be completed. After that, the polypeptide chain will be removed from the from the ribosome and moves towards its modifications, called as the post-translational modifications, which occur in the Golgi apparatus. Okay, now so this is an important point you have to remember, guys. The translational unit in mRNA flanked by a starting codon and the stop codon. There are some untranslated additional sequences in mRNA called as the untranslated regions, are the UTRs, present at both 5' end, that is the upstream or the before the start codon, and also on the 3' end. And UTRs are required for efficient translation, which means you can call them as the regulatory sequences which can control the translation. Clear? Right? Now this one, lac-operon. See guys, in the textbook it's mentioned that every expression of a gene is very tightly and properly regulated. One of the regulatory mechanisms explained in the textbook is of lac-operon found in the E. coli, or you can say in the bacterial cells, which was proposed by Jacob Monod. Jacob and Monod, right? So what is lac-operon exactly? Can you tell me, guys? In simple words, can you tell me how does it work? Lac-operon. Inducer, very good. What is that? You have the diagram in the textbook, right? Can you see? Look at this diagram here, everybody. This diagram. Where is the textbook? Huh? Can you see, guys? A lac-operon has a repressor gene, a promoter gene, operator gene, and structural genes. Correct? Now, in the absence of inducer, what is an inducer here? The lactose. Right? So there is no lactose. So simple, guys. Certain genes, they will not be transcribed and translated unless they are necessary. Now there is, the bacteria is living in a medium, there is no lactose. Is it necessary to activate this gene and produce an enzyme which is no more used? No, right? Which means in the absence of the lactose, the gene is inactivated. The inactivation comes from the gene called as the I gene, which produces an mRNA called as the repressor mRNA. So this repressor mRNA produces a protein called as the repressor. Now this repressor binds with the operator. When operator is inhibited, do you think the RNA polymerase can bind to it? No. So the gene is switched off. In the presence of inducer, this inducer bind with the protein. What is the protein here? Repressor, forming repressor inducer complex, and this complex is inactive and cannot bind with the operator. So as the operator is now free, the RNA polymerase can bind to it and continue the transcription of the Z, Y, and A genes, which can be translated to produce the 3 different enzymes necessary for the lactose metabolism. So this is called as this is how the regulatory mechanism works in the gene expression of the bacterial cells. Understood? Right? You have to go through it once, then only then you will understand. Now, the last part here, which I have to explain later, but I will see. Two topics are there. One is a Human Genome Project. The other one is, what is the other one, guys? The DNA fingerprinting. So Human Genome Project is only one objective, to find out the sequence of all the 46 chromosomes, which was launched in 1990, a 13-year project coordinated by US Department of Energy, the National Institute of Health, the Wellcome Trust in the UK, Japan, France, Germany, China. All of them are participated, and it was completed in 2003. And they found that human genome is approximately 3 into 10 to the power of 9 base pairs, and the cost of sequencing was about $3 per base pair, which is around 9 billion US dollars. And they used many non-human model organisms to amplify the genes like bacteria, yeast, and Caenorhabditis elegans, a nematode, a Drosophila, the plants include rice and Arabidopsis, right? Now, they've used 2 methods. One is the expressed sequence tags. What does it mean by expressed sequence tags? Focusing all the, identifying all the genes that are expressed as the RNA, whether it is tRNA, mRNA, or the RNA. Every gene is focused. Second one, sequence annotation. Blind approach of sequencing the whole genome containing coding and non-coding sequences. In the first one, only coding sequences. That is called as ESTs. In the second one, both coding and non-coding, and then joining them together using bacterial artificial chromosomes and the yeast artificial chromosomes. And what are the salient features? The first one, human genome contains 3,164.7 million base pairs. An average gene consists of about 3,000 bases. The largest gene is a protein called as the dystrophin, which contains 2.4 million bases. And the total genes estimated to be around 30,000. Almost 99.9% of nucleotide bases are exactly same in all the people. Functions unknown for almost 50% discovered genes. That means we have sequenced 30,000 genes, but we know the functions of only 15,000 genes. Less than 2% of the genome codes for the proteins. That means 98% of the sequence are the non-coding sequences. We do not know its function completely. Chromosome 1 has the most genes, that is about 2,968, and the Y has the least, 231. At 1.4 million locations, single base differences called as single nucleotide polymorphism are the SNPs. And SNPs can help in tracing the human history by comparing with the genes or genomes of the other organisms. Okay, now the extra information here. What's the next one, guys? Here, DNA sequencing. Sorry, DNA fingerprinting. Like we discussed just now, 99.9% base sequence among humans is same. And that 0.1% difference in sequence of DNA make every individual unique in their phenotype. We're all same in 99.9%, but 0.1% is different. And this DNA fingerprinting involves identifying the specific sequences differences in the repetitive DNA. So we all have the repetitive DNAs, but every repetitive DNA of every human is different. A small stretch of DNA repeated many times, which are called as the satellite DNAs. And depending on the base composition, length of segment and the number of repeated units are classified into microsatellites and the mini satellite, depending on the lengths. These repeated DNAs are known as the microsatellites and the mini satellites. Like you see, they do not code for any proteins. These are unique from person to person. And they form a large portion of the human genome and show a very high degree of polymorphism. That means they are repeated several times, several hundreds of times in a one person. And these are the repeated sequences which form the criteria of DNA fingerprinting, and we can collect them from any type of tissue like blood, hair follicles, skin, bone, saliva, or the sperms, but they show same degree of polymorphism, whether you collect it from the saliva or the semen or the hair follicles. Now, and these polymorphisms are inheritable from parent to child, so DNA fingerprinting solves majority of the paternity disputes. So how it is done? Simple. First, you isolate the DNA from the person, use enzymes called as a restriction in a nucleus to cut them into smaller fragments. Then separation of DNA fragments by using a method called as the electrophoresis, which separates the DNA fragments based on the length and the charge, right? And then you attach them onto a membrane called as the nitrocellulose or the nylon membrane. Then using variable number tandem repeats, we hybridize them using the VNTR probes. Then we detect the hybridized DNA fragments by a method called as the autoradiography, based on the patterns. And then you have a diagram in the textbook, the last section. Can you see the diagram, everybody, once? It's not in this chart. Can you see them, everybody? So we have two types of chromosomes here, the paternal chromosome and the maternal chromosome. What is this? Chromosome number 7. Next we have chromosome 2 and chromosome 16. If you notice in the next electrophoresis chart, that is mentioning amplified repeats separated by size on a gel give a DNA fingerprint. So what is the result in the last section here? Can someone tell me what is the result here? Look at the textbook. Where is the textbook? Huh? Tell me guys, what is the result? Anybody? What does it say? Where's the textbook, miss? Hmm? But I told that we'll discuss molecular basis today, right? In the last class? I told you. You were? Okay, tell me, result, everybody. So, read the statement in the figure 5.16. Schematic representation of DNA fingerprinting. Few representative chromosomes have been shown to contain different copy number of the VNTRs. For the sake of understanding, different color schemes have been used to trace the origin of each band on the gel. The two alleles, the parental and maternal of a chromosome, also contain different copy number of the VNTRs. It is clear that the banding pattern of DNA from crime scene matches the individual B, not with a A. So why is that? If you notice, the C1 here, the C. Let's say there is the fluid, body fluid we found in the crime scene, right? And we have two suspects, A and B. So A, B, which have been run together, you can see that the B chromosome pattern is very similar to the C, which means we know a suspect, right? And we have performed his DNA fingerprinting. We know the fluid from the crime scene. We performed the DNA fingerprinting. When we found the gel pattern on this machine, it's the same. Both of them are showing the same banding pattern, which means they have the same repetitive sequences. Means the person B is a, who is that? The culprit. Understood? Okay, so it is the DNA fingerprinting, guys. Are we clear? Yeah. So one chapter done. That is the molecular base of inheritance. Any doubts in this one? Everybody? Are we clear? All right. Now open the module and solve the questions. We have some time.